So now we're going to start focusing on the cell envelope, which is the layers, numerous layers that would be directly under the guide calyx. So just to kind of look at that, if we look at a prokaryotic cell, the outermost layer here is going to be the guide calyx. This particular bacteria has a capsule as a guide calyx, so it's more tightly bound. There is the option that it could have a slime layer, which would be more loosely associated with that. But if you look underneath the capsule, we have these numerous layers. There's actually 3 here. This is what's going to make up the cell envelope. So that's where we're going to be focusing on next. So with the cell envelope, it is made up of numerous layers. Some bacteria have 2 layers in their cell envelope. Some bacteria have 3 layers in their cell envelope. Regardless, though, the innermost layer, which is the layer that would sit right outside of the cytoplasm, which is fills the internal portion of the cell, the innermost layer is going to be the cytoplasmic membrane. You'll also hear me call it the inner cell membrane, potentially even the plasma membrane. You may see it in some of the images. But the cell membrane, or the inner cell membrane, is that initial layer that sits right outside the cytoplasm. Directly extending off of the cell membrane, there is going to be a cell wall. All bacteria have a cell wall. Gram-positive bacteria, their cell wall is typically a little thicker than what we would see in gram-negative bacteria. Now, if the bacteria are gram-positive, that's the only 2 layers they have, and we can see that over here. They would have the cell membrane, that's the inner cell membrane right here. So the cytoplasm and the inside of the cell would be here, and then the inner cell membrane would be surrounding that. And then directly outside of the cell membrane, they would have the cell wall, but note it's quite a thick cell wall. But that's the only 2 layers that they would have. Now, with gram-negative bacteria, it is different. Gram-negative bacteria actually have an additional layer. That additional layer is called the outer cell membrane. So if we look here, gram-negatives, in their cell envelope, they actually have 3 layers. The inner cell membrane, which we see right here, and then the cell wall. But note how much thinner the cell wall is for gram-negative bacteria as compared to gram-positive bacteria. And then only gram-negative bacteria get this 3rd layer right here. That 3rd layer is the outer cell membrane. So to summarize, gram-positive bacteria have 2 layers in their cell envelope, the cell membrane and the cell wall, whereas gram-negative bacteria have 3 layers in their cell envelope, the cell membrane, the cell wall, and then the outer cell membrane. So we're going to go over the basic structure and function of each one of these layers. So when we talk about the cell membrane or the cytoplasmic membrane, I do want you to know what it is composed of, which is the same thing that our cell membrane is composed of, and that's phospholipids. So the cell membrane, whether it's the inner cell membrane or the outer cell membrane in gram-negative bacteria, is basically composed of a bilayer or 2 layers of phospholipids. So here's a layer of phospholipids here, here's a layer of phospholipids here. They're arranged in this bilayer based on association with water, but it is a double layer of phospholipids. And then typically in the cell membrane, there are proteins embedded that can help with things like transport. So as far as the function of the cell membrane, its primary function is transport. So the cell membrane is involved in selective permeability. What that means is it controls what goes into and out of the cell. So it's going to control nutrients going into the cell and then waste leaving the cell. And that's kind of the standard function for cell membranes even in our cells. But with prokaryotic cells, their cell membrane has to do some heavy lifting. So in addition to controlling transport, prokaryotic cell membranes are also responsible for ATP production or energy production, which is different in eukaryotic cells. In eukaryotic cells, they are going to produce energy in the mitochondria. That's the powerhouse of the cell. But prokaryotic cells don't have mitochondria, so they actually produce their energy within the cell membrane. And then also nutrient processing. In eukaryotic cells, we have an organelle called a lysosome that has digestive enzymes that can process nutrients. But again, bacterial cells don't have that. That's a membrane-bound organelle. So they actually process their nutrients using enzymes found in the cell membrane. So the cell membrane is still involved in transport. That's its primary function. But with bacteria, it has multiple other functions, including ATP production and nutrient processing. So the cell wall. The cell wall is present for both gram-positive and gram-negative bacteria. But keep in mind, gram-positive bacteria would have a much thicker cell wall than gram-negative bacteria. So collectively, the cell wall, its primary structure or primary function is to maintain cell shape and support. So it kind of maintains that rigidity, that structure, support. I kind of think of the cell wall kind of like as a skeletal system, which obviously gives our body support and shape. If we took out our skeletal system, we would obviously just be a big ball of mush. Same thing with bacteria. That cell wall gives them that shape, that structure, support. If that is affected in any way, it can actually lead to the cell bursting through cell lysis. So the cell wall is really important for bacteria. I do need you to know what the cell wall is made up of, which is a chemical called peptidoglycan. Peptidoglycan is exclusive to bacteria. It's only found in bacterial cell walls. And we can see this up here. If this is the cell envelope of a gram-positive bacteria, and then over here we have the cell envelope of a gram-negative bacteria. So the cell envelope of a gram-positive bacteria is only made up of two layers. We have the cell membrane right here, and then we have the cell wall right here. But note how much extra peptidoglycan it has, which is what makes up the cell wall. And then over here with the gram-negative bacteria, in their cell envelope, they have the cell membrane right here, the cell wall right here, but note how much thinner the peptidoglycan layer is as compared to gram-positives. And then again, they get this additional outer cell membrane that only gram-negative bacteria have. The cell wall and its peptidoglycan layer are the target of different antimicrobial medications and enzymes. So for example, penicillin and the enzyme lysozyme, which is found in things like tears and sweat and saliva in our bodies, both of those chemicals work by breaking down the peptidoglycan layer in the cell wall. And when the peptidoglycan layer in the cell wall gets broken down, the bacterial cells can no longer maintain their structure and support. So again, they burst through cell lysis. So the cell wall and the peptidoglycan that composes it are very important for bacterial cells. So to kind of summarize some of the core differences between gram-positive and gram-negative bacteria. Gram-positive bacteria, we know that they have a thick peptidoglycan layer, so they would have a much thicker cell wall, whereas gram-negative bacteria have a thin peptidoglycan layer, so they would have a much thinner cell wall. Gram-positive bacteria, when you put them through a gram stain, which we previously learned about, they are going to stain purple. Gram-negative bacteria would actually stain pink if you did a gram stain on them. Gram-positive bacteria do not have that outer cell membrane. They only have two layers in their envelope, the inner cell membrane, cell wall. Gram-negative bacteria, though, have the three layers in their cell envelope, the inner cell membrane, the cell wall, and then that outer cell membrane. Something that we haven't talked about yet are the last column, or last row. Gram-positive bacteria in their cell wall have components called teichoic and lipoteichoic acid, and we can see those here. These would be teichoic and lipoteichoic acids, these orange and yellow circles embedded in the cell wall. What they collectively do is maintain all the extra peptidoglycan. Gram-positive bacteria have an excess of peptidoglycan, so they need these components to help maintain that. So that's the primary role of teichoic and lipoteichoic acid, is just to maintain the abundance of peptidoglycan in gram-positive bacteria. Gram-negative bacteria, they have these lipoproteins, which are fatty proteins, and lipopolysaccharides, which are fatty sugars, in the outer cell membrane. So if we look at the outer cell membrane, we can see some lipopolysaccharides here in the yellow and some lipoproteins here in the brown, but they're embedded there in the outer cell membrane. And what they do is they act potentially as toxins. So what can happen is bacteria that cause infections, if they're gram-negative, they can actually release these lipoproteins and lipopolysaccharides into our body, and that can cause a variety of different things. It can cause us to go through vomiting, diarrhea, muscle spasms, potentially paralysis, all different types of things. So lipoproteins and lipopolysaccharides we'll learn about them later on in the semester, but they can be quite toxic to our body. We already went through the gram stain once, but just to go over it again, repetition is our friend in this course. We are going to look at the reagents and kind of look how everything works. But with the gram stain, what you're trying to do is differentiate between gram-positive and gram-negative bacteria. And again, the primary difference is that thickness in the cell wall. So do make sure you know the reagents in the order that they're used in. So our first reagent is crystal violet. It is going to stain all the bacteria purple, regardless of whether they're gram-positive or gram-negative initially, because it's the primary stain, it's the only one you've used. Then we use iodine. Iodine is not going to stain, but it is going to act like a sealant. It's a mordant, so it's going to seal the crystal violet color in, and everything's still purple. Then we rinse with alcohol, which is the most important step. This is the decolorization step, and it is what's going to separate gram-positive bacteria from gram-negative bacteria. When you rinse with alcohol, the gram-positive bacteria will stay purple. They have a thick cell wall, so they are able to hold on to the crystal violet even when rinsed with alcohol. The gram-negative bacteria, when you rinse them with alcohol, it dissolves their outer cell membrane, and then the crystal violet is only weakly held on by the very thin cell wall. So the alcohol is actually able to pull the crystal violet out of the gram-negative cell wall, and they will temporarily be clear. Our very last step is to counterstain with a dye called safranin. Safranin is a pink dye, and anything that lost its color in the decolorization step is now going to stain pink. So in the end, gram-positive bacteria will be purple, and gram-negative bacteria will be pink. It's also important to consider if potential mistakes were made. So let's say we forget to use iodine. We do crystal violet, immediately rinse with alcohol, and then try to stain safranin. Well, if that's the case, the crystal violet is not going to seal in. So when we rinse with alcohol, we're going to pull the color out of all the bacteria, even the gram-positive. And in the end, everything is going to stain pink. Let's say you were going through the procedure and you forgot to use alcohol. So you just did crystal violet, you sealed it in, and then you went straight to safranin. Well, if you don't rinse with alcohol, you're not going to pull the purple out of the gram-negatives. And the purple will stay, or the gram-negatives will stay purple just like the gram-positives. So in the end, everything would just appear purple. So it's very important that you stick to the proper order of the reagents, which would be crystal violet, iodine, alcohol, and then safranin.